Hello and welcome back to the Biochemistry for Health Sciences channel. In today's video, we shall discuss the executioner caspases. Now, in previous videos, we saw how caspases, the initiator caspases such as 8 and 10 from the extrinsic pathway lead to the activation of executioner caspases. And then we also saw from the intrinsic pathway how the initiator caspase 9 activates the executioner caspases. So today we'll talk about the actions of these executioner caspases. What do they do? By the way, cleave caspase 3 is actually used as a marker of caspase activity and apoptosis activation. So in research, when we want to see whether the cell is undergoing apoptosis, detecting this cleave caspase 3 is one way of doing that. Okay, so once these executioner caspases are activated, what do they do? Now recall the substrate for these enzymes caspases are proteins. So we have a protein substrate for the executioner caspase. So whatever the protein substrate is, the caspase will act on the protein substrate, cause a proteolytic cleavage, and that might make this protein inactive, or it might make this protein active by removing some inhibitor of this protein, maybe some other protein that inhibits this protein. The caspase could destroy this protein and make this protein active, or there could be a domain here in this protein that is inhibitory, so the caspase could remove that domain and make this protein active, or the breakdown of the protein may have no effect at all. The protein is simply cut up and destroyed. So let's see some of these actions of executioner caspases, what they do to the cell to cause apoptosis by acting on certain protein substrates present in the cell. Now, one of the effects of the executioner caspase is the destruction of the nucleus. Executioner caspases, especially caspase 6, cleaves proteins called laminin AC. These are proteins that provide structural support to the nucleus and also the interaction of the chromatin with the nucleus. So by destroying these proteins, the chromatin starts clumping together, what we call pyknosis, and the nucleus starts fragmenting into fragments, nuclear fragments, that form small apoptotic bodies. Now, cleave laminin AC is a marker of caspase activity as well as apoptosis activation. Now, another effect of this executioner caspases is DNA fragmentation. So what happens here is this executioner caspases destroy, cleave an inhibitor of this enzyme called caspase-activated deoxyribonuclease, or CAD. So there is an inhibitor of CAD. Now, so these caspases destroy this inhibitor of CAD, what we call ICAD. So when this ICAD protein is destroyed, this CAD becomes active. And now this CAD will cleave those DNA regions that are exposed. These are the regions between nucleosomes. So for example, this part here shown in arrows, this CAD, this deoxyribonuclease will start cleaving this DNA around here. And this will release these fragments out. So you can see this will come out, this will come out, this will come out. So these are about 200 bases DNA fragments. And these fragments, when you run a DNA electrophoresis, they look like a DNA ladder. You see, they're difference of 200 molecular weight, 400, 600, 800, and so on. Okay, so this is what we see when we see these cells in culture undergoing apoptosis. When you run a DNA electrophoresis, this is a sort of a pattern that you see because of the action of executional caspases. Another effect of these executional caspases is that they prevent DNA repair by this protein called PARP1, which is poly-ADP ribose polymerase 1. Now, PARP1 plays a very important role in DNA repair. So what happens is, and we'll talk about PARP1 in another cell that called parthenators. We'll talk about this in a future video. But by destroying PARP1, it prevents the cell from repairing the DNA that has been cleaved. In fact, cleaved PARP1 is also used by researchers to see whether the cell is undergoing apoptosis and whether there is a caspase activity that's going on. So by detecting cleaved PARP1, that tells us the caspase is active and the cell is undergoing apoptosis. Now, another effect of these executional caspases is that they release these LPCs, what we call lysophosphatidylcholine. These are find me signals. So it basically releases these signals to tell immune cells, the phagocytes, to come and find this cell that is undergoing apoptosis. So executional caspases, they activate this enzyme called phospholipase A2, PLA2. And what PLA2 does, it removes one of the fatty acids. So it removes one of the fatty acids from this phosphatidylcholine. As you know, this is one of the fatty acids, one of the amphiphilic lipid found in cell membranes. So that forms LPC. And this LPC is released by the cell that's undergoing apoptosis, by this dying cell. And LPC is a find me signal. It helps attract the macrophages. So this LPC that is released by the dying cell will bind receptor on the macrophage, such as the GPCR-G2A receptor. So this interaction will bring the macrophage towards the dying cell. There are other find me signals, examples ATP, AMP, UTP. So the dying cell will release these find me signals. And how is that done? So executional caspases, they cleave the C-terminal part of this protein, connexin like pannexin 1 channel. And by cleaving this part here that blocks the channel, the protein channel opens up. And now these nucleotides leave the cell. Okay, so the channel opens up, these nucleotides will be exported out of the cell, and once they get out of the cell, they will bind these receptors, these GPCR receptors on the macrophages called P2Y, purinoreceptors. There are several of these receptors, so this binding will now attract the macrophage towards the cell that is dying by apoptosis. Now, besides find me signals, there are also what we call eat me signals. These are signals that tell the macrophage that I'm a cell that's dying, eat me up. So these are eat me signals. Now, one very important eat me signal is phosphatidylserine. And we talked about this under amphiphilic lipids. So phosphatidylserine is an important eat me signal. So in healthy cells, we have enzymes such as flippase, and what they do is any phosphatidylserine, and you can see that here in pink, any phosphatidylserine that is on the outer leaflet of the plasma membrane, they are flipped. They are sent to the inner leaflet. Okay, so there is no phosphatidylserine that is exposed on the outside of a healthy cell. Okay, so all the phosphatidylserine is inside, facing the cytoplasm of the healthy cell. We also see other amphiphilic lipids such as phosphatidylinositol and phosphatidylethanolamine. These are also present mostly on the inner leaflet towards the cytoplasm. Okay, so healthy cells do not have phosphatidylserine facing the outside of the cell. So what do executional caspases do? They cleave and destroy the flippase enzyme. So now this flippase enzyme cannot take this phosphatidylserine and send it into the inner leaflet. There's another thing that these caspases do. They activate other enzymes called scramblases. Now scramblases do the opposite. Okay, they actually take the phosphatidylserine from the inner leaflet and send it to the outer leaflet. So by inactivating flippases and activating scramblases, we see a lot of phosphatidylserine on the outside of the dying cell. So this phosphatidylserine on the outer leaflet tells the macrophage to eat the cell. It's an eat me signal. And there are receptors on the macrophages such as the TIM receptor, which stands for T cell, immunoglobulin and mucin domain containing molecule. These phosphatidylserine bind to these TIM receptors, and this macrophage will now get attracted, it will be bound to the cell, and it will start eating the dying cell. Another effect or action of these executional caspases is that they cleave several cytoskeletal proteins. Remember, cytoskeletal proteins are proteins that give the structural integrity to the cell. So by cleaving proteins such as actin and vimentin and so on, the cell loses its structural integrity. Now, another effect of these executional caspases is they help form membrane blebs and apoptotic bodies. The exact mechanism is not clear, but here is one possibility. What happens is these executional caspases, they cleave by cleaving these proteins called Rho-associated kinase 1 or ROC1. They activate these proteins. And this ROC1 then phosphorylates myosin light chains. Okay, myosin light chains. And there is just under the plasma membrane, there is this layer of actin-myosin cytoskeleton. Okay, so there is this layer just below the plasma membrane. So by activating the myosin light chains, that will cause the myosin light chains to contract, and this layer below the cell start contracting. They start contracting. This contraction will decrease the volume of the cell, and as the volume goes down, the pressure in the cell increases. And this causes the detachment and the expansion of plasma membrane from the underlying actin cortex. And that leads to, this detachment leads to the formation of the bleb. Now, as this happens, the actin myosin cortex, cytoskeleton cortex, reassembles below the plasma membrane to stop the bleb formation. But once again, the actin contracts because this caspase 8 is, caspase 3 is activating this ROC1. So once again, there's this contraction and this continues. So the expansion and contraction of blebs leads to the packaging of cellular debris into the apoptotic blebs, which later form apoptotic bodies. So by contraction and expansion, these things that are being broken down in this dying cell, they are packaged into apoptotic blebs later that form apoptotic bodies. Okay, so eventually the cell, as this goes on, the cell contracts and shrinks, forming apoptotic bodies. So what you see in an apoptotic cell because of these executional caspases, we see morphology, for example, the cell shrinks. The plasma membrane is intact, especially in the early and mid stages. There might be a rupture of the plasma membrane in the later stage. However, the plasma membrane is altered. Remember, for example, the phosphatidylserine is present on the outside. Then we see chromatin clumping, the fragmentation of the nucleus. We see a lot of formation of apoptotic bodies. So these are debris that are surrounded by a membrane. Okay, so apoptotic bodies. And then we see also a lot of blebs. See the bleb formation as we discussed. And under a microscope, if you see this, you will see in vivo a lot of phagocytic cells such as macrophages that are being attracted to this dying cell and are ready to eat up the apoptotic bodies as well as eating up the dying cell. Okay, so the end result is these phagocytes will eat the dying cell and all the apoptotic bodies, and that's how the cell is eliminated. And remember, in this elimination, there is no inflammation. It's simply the cell being eaten up by the phagocytes. No inflammation because the plasma membrane is intact, so there's really no loss of cellular content. Okay, it's not like biomolecules from the cell getting out of the cell and going into the extracellular fluid and so on. So there's no inflammation. So that's one thing nice about apoptosis. It can destroy cells, eliminate cells without triggering inflammation. Okay, so that is all on apoptosis. In the next video, we shall discuss necroptosis. So until the next video, once again, thank you for watching. Have a wonderful day. We'll see you in the next video.